Hello, everyone. My name is Raika Karimi. I'm a master's student at Concordia University. Today, I'm going to explain our paper on behalf of our team. The name of the paper is Deep Video Canonical Correlation Analysis for Steady-State Motion Visual Evoked Potential Feature Extraction. This paper is going to be published in European Signal Processing Conference. Before starting, let's take a look at the outline. We will provide an introduction to brain-computer interface. We will describe different sorts of visual evoked potential and new emerging paradigms called steady-state motion VEPs. Then we illustrate our experimental protocols, our own designs of SSMVEPS paradigms, and signal processing modules used in our experiments. We'll elaborate on the architecture of deep video canonical correlation analysis proposed in this paper. Finally, we compare the result obtained in the experiments with other deep learning-based counterparts. There is a question that whether interactions between our brain and the outer world can be established without intervention of humans' voluntary nervous system. Attempts toward achieving this goal has resulted in the emergence of brain-computer interface systems. If we look at the figure at the right top, we can understand the goal of BCIs. EEG signals are acquired when stimulation are shown to the subjects, and the collected signals are translated to commands used to control the outer devices. There are a lot of applications for these systems. For example, one of them is neuroprosthesis, which is a device to overcome neural disabilities caused by injuries. Another example is P300 Speller, which is a system developed so that users can input text to computer through brain signals and without using their limbs. Neurogaming, neuromarketing, and so on are other applications of BCI. One of the non-invasive BCI is visual evoked potential-based systems. According to the figure at the right bottom, displaying visual stimulus to the subject leads to provocation of signals within the visual cortex. Once we collect these signals, we can extract pattern change after watching the stimulus. These features in EEG signals are called event-related potentials, or ERP. We have different components of ERPs, including P1 or P100, N1 or N100, and P300, which are the most important components of ERP. One of the most important VEPs are steady-state VEPs, which is called SSVEP. SSVEP is a response to a steady-state visual stimulus, modulated usually at a frequency higher than 4 Hz. And different frequencies of flickering are assigned to different targets or commands in these systems. Targets are onset offset patterns appearing with constant frequency. Black and white filtering is one example of these paradigms. SSVEP response is characterized by an increase in amplitude at stimulus frequency, resulting in significant fundamental and second harmonics at the occipital region. Steady-state visually evoked potential, or SSVEP, has become popular due to their fast and easy system configuration and a strong provocation of visual brain signals. Despite the popularity, their utilization for practical applications, especially for assistive technologies, is challenging due to eye fatigue and risk of induced epileptic seizure. The uncomfortable flicker bias modulation of existing methods restricts practical interactivity of BCI application. In this regard, the issue is addressed by development of flicker-free steady-state motion visual evoked potential, or SSMVEP. For example, in the contraction expansion of checkerboard patterns like figure A, each ring switches the colors of its black and white board, and all the rings do this action respectively in such a way that the subject imagines that the image is contracting and expanding, which means the movement starts from the center of images and goes to the border. We call this state expansion. And then it comes back to the center, and we call it contraction. Frequency of motion direction change is equal to the frequency of SSMVEP. Swinging, rotation, contraction, expansion, Newton rings, and radial zoom are kinds of SSMVEP stimulation. With regard to the SSMVEPs, there is a major problem. SSMVEPs are not as accurate as regular SSVEPs in terms of target identification and provocation of the VEP signals. Therefore, in this research, we come up with 4 SSMVEP paradigms as well as deep learning-based target detector to address this deficit of them. To remove the unwanted signal components, first, zero-phase Chebyshev type 1 filter, which is a bandpass filter between 2 to 40 Hertz, are applied to the raw EEG signals. The goal of the filtering is to smooth the data and remove high-frequency artifacts. This frequency interval is determined using the visual frequency band of the human. Apart from the time filtering, we provide spatial filtering, which is a common approach in brain-computer interface. To use topological distribution of signals to remove the components within EEG that are unnecessary for main goal, which is extracting VEPs in our case. To that end, instead of using one electrode, we incorporate 9 EEG channels, mainly located at occipital and parietal lobes. We also have frequency recognition, aiming to classify different signals or identify target frequency within the EEG signal. How does basically CCA work to label an unknown signal and detect its susceptible target frequency? So the CCA is one of these frequency recognition algorithms, and we want to understand this mechanism. We have 2 data sets called observation and reference signal. And the reference signals include sine and cosine signals oscillating at SSMVEP stimulation frequencies. And we aim to find projection vectors leading to the largest correlation coefficient between projected data sets. For each stimulation frequency, the correlation coefficient is calculated. And the final label is the stimulation frequency for which its correlation coefficient is maximum. In this figure, we can observe the mechanism of our visual BCI system from telecommunication point of view. There is an analogy between these 2 fields. Discrete information are assigned to the different targets displaying on the screen. Targets are modulated with different frequencies. These videos play the role of the transmitter in our models. So we will come across a frequency modulation light signal in the visual receptive field. And emitted photons will be transformed to firing neurons within retina. Then the signal is passed to the visual pathways of the brain. Ultimately, the modulated signal reach visual cortex where the most of EEG electrodes are implanted in our experiment. These electrodes correspond to the receiver. The visual pathways, retina, and visual receptive field all will be part of a channel. This channel is obviously not an AWGN channel, and a nonlinear and the complex system need to be used to model and estimate the changes applied to carrier signals. Because the brain is one of the most nonlinear and complex systems due to existence of activation function, other intrinsic characteristics derived from the structure of the brain, we need to remove these facts from the signal. The question is how to model the nonlinearity of the brain's visual pathways referring to a channel. To address this problem, we come up with deep learning-based algorithm because they act like a nonlinear function. The input is noisy and modified signal, and the output are new embeddings of signal. What if we determine the nonlinearity in such a way that embeddings are compatible with the models used for ideal linear systems and alleviate nonlinearity? The best way to find these nonlinear functions is to use algorithms fitted to the template and training sets of the same problem. They automatically learn how to remove these biases, noises, and the nonlinearity from the collected data. The CCA is an unsupervised technique. In this section, we want to introduce a supervised classifier for target identification task. We deploy a deep learning-based design consisting of 5 fully connected dense layers. As the color of a pixel of a video changes during the time, a potential will be evoked in the EEG signal. Our goal is to find superposed VEPs caused by all pixels. Eccentricity, which is the distance of the pixel from focal point, has high impact on the intensity of the evoked signal. The focal point is the point where the subject has to stare at. The further the movement is from the center of attention or focal point, the less it has impact on VEP. As it is shown in the picture, we group the pixel of each RGB layer based on their eccentricity. Here. In other words, those pixels which have the same distance from the focal point are located in the same group. Time signals corresponding to pixels in each group are averaged to generate a tensor Z, which has 3 dimensions. C is the number of group of pixels, and the Z matrix is fed to 3 independent, fully connected neural nets, which are main parts of video feature extractor called VFE layer. Apart from VFE layer, we have 2 types of nonlinear projection. One for datasets derived from the videos, the output of the VFE layer, and another one is dataset corresponding to the EEG signals. The objective is to project 2 datasets, X and Y, to a new space where the linear CCA is compatible to the embeddings, the new embeddings of 2 datasets. Matrix X, with the size of K times M, represent the EEG time samples from K channels, and this is the input of the first segment. Matrix Y, with the size of 3F times M, where M denotes number of time samples and 3F denotes output size of the video feature extractor, is the input of the second segment and output of the VFE layer. The propagation rule of each segment with input X is defined as follows. We can see it here. I don't know if the cursor is visible or not. For SSMVEP paradigms are designed for the experiment, including reciprocal rotation, radial zoom, swing, and sway. The motion modes are represented in this figure. 5 men and 5 women between age 20 to 27 participated in this test. The viewing distance from monitor is 70 centimeters. Subjects participated in the test for 4 rounds with a 10-minute break between the rounds. In each round, all targets are displayed one by one, and subjects are asked to look at the target where the cursor is pointing at. Experiment is repeated 2 times for each target, and we have 4 trials for each one. Each trial lasts 3.5 seconds, and there's a 2.5 seconds break between 2 consecutive trials. Here we can see the video of the test. User-specific training as well as eightfold cross-validation are performed for training and evaluating the methods. If we look over diagrams in this slide, we can understand that swing paradigm achieved the best accuracy of 96.8% and the information transfer rate of 35.2 bits per minute. The ITR is a criteria to measure the speed of our BCI system. By focusing on each column of these diagrams, you can understand that DVCCA outperforms its CNN-based counterpart in terms of the accuracy and the ITR. And for all of the paradigms, DVCCA outperforms other neural network-based algorithms. This model also avoids overfitting. It doesn't need a large training data set. As we mentioned in the architecture description, the model provides more interpretation rather than CNN for understanding the mechanism of classifier. As a conclusion, the proposed DVCCA model consists of a video feature extractor or VFE layer that uses characteristics of video to independently fit to the template EEG signals of each individual. Therefore, the extracted features from EEG signals are more correlated with the stimulation video signal. The proposed model addresses problems associated with more complicated networks, such as overfitting or lack of enough training data. Finally, most of the neural network-based classifiers used to identify SSMVEPs are black boxes, and there is little evidence of interpretation behind the superior result of their frequency identification modules. We try to integrate stimulation characteristics and intrinsic features of the brain signals into our deep learning-based model to provide some intuition behind the model architecture. Finally, I want to thank other teammates and my supervisors who have contributed to this paper. Thank you for your attention, and please feel free to join the Q&A session for the further questions.